Welcome back to Chapter 7. We're moving on to Part 2, where I'm going to give you a brief introduction to cellular respiration and some in-depth information on glycolysis. So as an overview, there are three parts of cellular respiration. There's glycolysis, and this is where glucose or something else, but again, we're focusing on glucose, is broken down into two molecules called pyruvate. And all of this happens in the cytoplasm. The next stage is pyruvate oxidation and the citric acid cycle. In these stages, we have a complete breakdown of glucose. This happens in the mitochondrial matrix, and at the end of this, all the carbons from the glucose molecule are changed into molecules of carbon dioxide. The third stage is oxidative phosphorylation, and it's in oxidative phosphorylation where we have the vast majority of ATP synthesis. This occurs at the barrier between the matrix and the intermembrane space of the mitochondria. Remember that mitochondria have two membranes, the inner membrane and the outer membrane. So it's happening right there at the inner membrane, and takes advantage of those two spaces, the matrix and the intermembrane space. Your book does a fantastic job of using colors to indicate which stage of the cellular respiration we're in. So this blue color here is for glycolysis. Orange, either light orange or dark orange, is going to be for pyruvate oxidation and citric acid cycle. And purple is for oxidative phosphorylation. All three of these stages are involved in harvesting energy from glucose. So a quick overview, looking at those three stages and learning a few more terms here. So the first one is glycolysis. And what happens in glycolysis, again, is that glucose is broken down into pyruvate. And this happens in the cytosol. And in glycolysis, we have something called substrate level phosphorylation, which I'll give you a few more pieces of information about later. But remember that the word substrate is always referring to an enzyme. It's the reactant of an enzyme. So the next stages are pyruvate oxidation and citric acid cycle. With pyruvate oxidation, we take the two pyruvates that are formed from glycolysis. There's two of them, and those go into the matrix, and they're oxidized into something called acetyl-CoA. Acetyl-CoA enters the citric acid cycle, and from this we get NADH and FADH2. Both of those are electron carriers and coenzymes. I forgot to mention earlier that we do get some NADH from glycolysis. We also get substrate level phosphorylation from this as well. So that's what happens in pyruvate oxidation and the citric acid cycle. The next thing that we're going to talk about is oxidative phosphorylation. And with oxidative phosphorylation, we have two main things that are happening: the electron transport chain and chemiosmosis. That's the word right there, chemiosmosis. It's a little hard to say, but once you get the handle of it, it's not too bad. The electrons from NADH that happened in both glycolysis and in citric acid cycle go to the electron transport chain and release their electrons in the manners that we talked about before, a whole bunch of redox reactions. And in this process, we get something called oxidative phosphorylation. So we're phosphorylating ATP, but it's happening, instead of at substrate level, it's happening with this process called chemiosmosis. And oxygen absolutely is going to be playing an important role in that as our final electron acceptor. So there are a few stages of cellular respiration again, and one of the most confusing ones is oxidative phosphorylation. This is a very mechanical process. And I just have a quick cartoon here for you. Don't worry about the words on here right now. We are going to be learning all of those words. But for now, just take a look at the picture. Oxidative phosphorylation accounts for 90% of the ATP that's generated by cellular respiration. And what we're doing is we're adding inorganic phosphate to ADP to make ATP. But you can see that it's a very complicated process. And there's lots of movement of things across the membrane. We're moving protons into the intermembrane space, and then we're going to allow those protons to move back through following their concentration gradient. And when that happens, that's when we make ATP. That's oxidative phosphorylation. The next one that we're going to take a quick look at is substrate level phosphorylation. And with substrate level phosphorylation, again, we've got some enzymes going on here, right? So we're going to make a small amount of ATP by removing a phosphate group from a substrate and transferring that to ADP in order to make our product, of course, and ATP. And that's going to happen a number of times, not a lot of times, but definitely a few times through the process of glycolysis and citric acid cycle. When you go through the entire cellular respiration, you're going to make approximately up to 32 molecules of ATP, and you'll also have our byproduct of CO2 and water. So remember to keep that in mind that's like the big picture thing that we're trying to keep track of a lot of stuff in this lecture. And carbon dioxide, water, and ATP are absolutely things that we're going to be keeping track of. So let's dive into glycolysis. This is where we're going to start to get some really big details and really careful details about what happens to glucose and how energy is harvested. And the end product of this is, of course, pyruvate. Literally, glycolysis means sugar splitting. Lysis, we've heard that word before, like hydrolysis, and we've heard of lysosomes. So glycolysis means sugar splitting. We're breaking down glucose into 2 molecules of pyruvate. And we're also going to make a couple of water molecules as a byproduct. Pay attention to those as we're going along. Glycolysis occurs in the cytoplasm and has 2 phases. There's the energy investment phase. In the energy investment phase, we're going to be using up a little bit of ATP. And then we have the energy payoff phase. And in the energy payoff phase, we're going to be making a bunch of ATP, actually 4 total. In the energy investment phase, we're going to be using up 2. 4 minus 2 is 2. So that's why we say that the net yield of the energy, of glycolysis is 2 ATP. And then we get some NADH as well. So glycolysis occurs even if there's no oxygen around. You're going to have glycolysis. If oxygen is present, the energy from pyruvate and the energy in the NADH is going to be extracted in the process of pyruvate oxidation, the citric acid cycle, and in oxidative phosphorylation. I just wanted to take a minute to remind you that we are using the mastering program online, and this has a lot of amazing animations that I strongly urge you to go see. We're going to be covering a lot of material, but it's very static, and since we're talking about a process here, it's nice to have an animation that covers what's actually going on in a step-by-step process in an animation. So, but we are going to move on with the lecture, and let's take a closer look at glycolysis. I just told you that there are 2 phases, the energy investment phase and the energy payoff phase. And I mentioned how much ATP is going to be important in each one. So in the energy investment phase, we are going to use up 2 ATP, and we'll have those ADP and inorganic phosphate as byproducts. In the energy payoff phase, we're going to have 4 ADP and phosphates joined together to make ATP. We're also going to be harvesting those NADHs as well as our pyruvates and some water. So if we add everything up together, we're going to have 2 pyruvates and 2 waters, 2 total APPs, because 4 minus 2 is 2, and 2 NADHs plus 2 protons. So keep that in mind as we get into some of these lovely details. Okay, so there are many steps to glycolysis. I'm showing you the first half of the steps right now, and I'm going to go into these details with you. So maybe take some really careful notes here. I'm going to go into these details with you in a step-by-step fashion. This is just the overview of the first half of glycolysis. Notice that we have right here and right here ATP being used up and ADP as a byproduct. And also notice, very interestingly, what happens to the phosphate isn't that it becomes inorganic phosphate. It actually becomes part of this molecule right here. And when we use it up again, we're going to put the second phosphate onto this molecule right here. Then we split that molecule into 2 parts. So let's go ahead and take a look at those in detail. So we're starting out with glucose. And glucose, in the presence of hexokinase, which is an enzyme, remember kinases they deal with the phosphates, right? So in the presence of hexokinase, we're going to take glucose and we're going to add a phosphate, stealing a phosphate from ATP. And we're going to add that phosphate to glucose to the 6th carbon of glucose. So now, instead of it being called glucose, we're going to call it glucose 6-phosphate. It's still glucose, it just has an extra phosphate on there. This is a little misleading here, but it's okay. We're just not showing all the hydrogens and oxygens there. So hexokinase takes our 6-carbon glucose and adds a phosphate molecule to that to make it glucose 6-phosphate. The next thing that we're going to do is take an enzyme called phosphoglucose isomerase. Remember that isomerase is a root of isomer, and isomer just means to change the way that the molecule is organized. So an isomer is a different version of it, but has everything else as the same. In this case, we have glucose 6-phosphate becoming fructose 6-phosphate. It still has the same molecular formula. It's just fructose instead of glucose, slightly different organization. After we make fructose 6-phosphate, we're going to use an enzyme called phosphofructokinase. Remember, we got a kinase again, and whenever we have a kinase, we're going to take a phosphate, at least in glycolysis, we're going to take a phosphate and we're going to put it on a substrate. So in this case, the phosphate is being put on fructose 6-phosphate, and it's going to become fructose 1,6-bisphosphate. The word tells you everything. The phosphates are on both the first carbon, which is right here, and the 6th carbon, which is right here, fructose 1,6-bisphosphate. Bis just means 2, so there's 2 phosphates on there. The next thing that we're going to do is we're going to split fructose 1,6-bisphosphate into 2 parts, and that happens in the presence of aldolase. Aldolase that cuts the sugar molecule into 2 different 3-carbon sugars. So the first sugar is called glyceraldehyde 3-phosphate. Glyceraldehyde is a 3-carbon sugar, and it has phosphate on the 3rd carbon. And the second one is dihydroxyacetone phosphate, or DHAP. This one is called G3P, by the way, and G3P is going to play an important role when we get into photosynthesis. So kind of try to remember G3P. But we have G3P and we have DHAP. And we do actually have a conversion that goes back and forth between DHAP and G3P. It never really reaches equilibrium because the thing is that only G3P is going to be used in the next phase. So here we are with G3P, and it is entering into the second half of glycolysis. G3P is going to have some hydrogen removed from it in the presence of an enzyme called triose phosphate dehydrogenase. So G3P is oxidized, and the electrons are transferred from NAD plus to form NADH. And then we're going to use the energy from that conversion. We're removing the electrons and removing that hydrogen from the G3P. And that is going to allow us to add in another inorganic phosphate. So what that makes is 1,3-bisphosphoglycerate right there. And 1,3-bisphosphoglycerate is going to then go through the process of now harvesting some of this energy into ATP. We did harvest some energy here in the form of NADH, and now we're harvesting some energy here in the form of ATP. We're taking that phosphate off of the 1,3-bisphosphoglycerate, and we're in the presence of phosphoglycerokinase, and we're going to make 3-phosphoglycerate. So 3-phosphoglycerate in the presence of phosphoglyceromutase, we're just reorganizing that, and we're putting the phosphate in a different location. So instead of it being 3-phosphoglycerate, it's now 2-phosphoglycerate. Remove a couple of water molecules from there, and you make something called PEP, phosphophenol pyruvate. When we remove that phosphorus from phosphoenol pyruvate in the presence of pyruvate kinase, we're going to make pyruvate. Here's our second location where we harvest energy from this process. One thing I'd like to mention to you again is that there's two separate molecules of G3P going through the process of the second part of glycolysis, the energy investment stage of glycolysis. So the way that your book shows this is that it shows like two little pieces of paper right here, and you can see that kind of concept is followed through this, and it even has the number 2 there for you. So just kind of keeping in mind as we're doing the accounting process that there are 2 molecules of G3P that go through this. We end up having 2 molecules of pyruvate in the end. Well, that's what I've got for you for Chapter 7, Part 2. We are about to move into Chapter 7, Part 3, where we get the details of pyruvate oxidation and the citric acid cycle.